So this lecture is going to cover bacterial genetics. This lecture starts off with a little review over DNA, kind of what DNA is, how it's put together, how RNA is formed from DNA, the central dogma of molecular genetics. DNA is used to make RNA, which is then used to make proteins, and then how that RNA is used to make proteins, and then how the proteins are put together, and how you read a piece of mRNA to make a protein. And most of that's review. I'm going to skip over that stuff and post a video review online for you guys about that kind of stuff. So I'm going to skip over a lot of the introduction stuff. A lot of that you guys have had this for since from biology 1, maybe some other science courses. So like I said, I'm going to go ahead and skip over most of the introduction stuff. So this is the stuff about what DNA is put together, how it's put together in here. How DNA works, how it replicates in bacteria. And like I said, I'm going to post some videos for you guys to see how this stuff works, the different types of RNA. A lot of this stuff is review, how proteins are made, how you read a codon chart. Like I said, I will post videos about how translation is made, how transcription works, how proteins and mRNA are made, and things like that. The concept of introns and exons, I'll post videos about this stuff. What an operon is, like I said, I will not leave you hanging on this. I will post some extra videos, some extra supplemental stuff for this, but a lot of the DNA replication information and things like that are just simply review. So let's go ahead and start with the actual microbiology portion of this lecture. So let's go ahead and jump into the stuff that's exclusive pretty much just for bacteria. So like I said, I will post a couple of videos for you guys, some supplemental information about the DNA replication, transcription, translation, codon charts, and things like that, since most of that stuff is usually review. If it's not, that's okay. Like I said, I'll post the information for you guys so you will have something to look at. So let's go ahead and start with the actual bacterial genetics. Genetics, once again, is being the study of DNA and how it's passed from generation to generation. So this guy right here was kind of the godfather of bacterial genetics. He's kind of the guy that kicked this whole thing off, Frederick Griffith. He did a very, very, very famous experiment using a bacteria called Streptococcus pneumoniae, which was previously known as pneumococcus, or pneumococcal pneumonia is the disease that it causes. So pneumococcal pneumoniae was the previous species that it was known as. Well, anyway, he noticed when he looked at this bacteria underneath in his lab underneath a microscope, that it came in two forms, just naturally speaking. This is natural variation. This is evolution. This is just how nature works. Things are different. You can be the same, but not the same. So that's kind of how that works. Like humans all have different hair color, eye color, same, even though we're all humans. Anyway, so the two different forms of the Streptococcus pneumoniae bacteria that he noticed underneath his microscope, one of them had a capsule and one of them didn't. Now, if you recall, the capsule on bacteria allows them to pass through the phagocytosis, the white blood cells that allows them to become immune to that. But this particular form of the bacteria would survive within the bloodstream and was able to cause disease, where the unencapsulated version was not. The immune system was able to keep it in check. It could not cause disease. So if you didn't have the capsule, you couldn't cause disease. If you had the capsule, you could, and that's exactly what he noticed here. That's the basis of his experiment. Now, he wanted to figure out what the difference between the two cells were. What was causing this disease? Was it the capsule or something like that? He wanted to know. So he did a very interesting experiment. So he took this one, he termed these two strains, by the way. The rough strain, the cell that lacked a capsule, that was totally unable to kill mice. And he termed the other one the smooth strain, the strain that had that capsule. So that's that we're going with, the rough and the smooth strain. So what he did was he took this rough strain of bacteria, and he put it in a syringe and he injected it into a mouse. He took a little while for this mouse to do its thing, and then he killed the mouse and took an autopsy on the mouse after the mouse a couple of days later, and the mouse was perfectly fine. It never developed any symptoms of pneumonia or anything at all. He did an autopsy on this mouse, a necropsy, and found this form of the bacteria inside of that mouse. Makes perfect sense. This bacteria, this form, is unable to cause disease. The presence of the capsule is what makes it cause disease. He didn't know that yet. So this is what's going on here. It doesn't kill the mouse. The capsule it lacks, so it can't cause disease. So if the mouse never develops any symptoms, and then when he did a necropsy on it, he just found this bacteria inside, which makes perfect sense. So the next thing that he did was he took that smooth strain, the strain that had that capsule around it, and he injected that into a different mouse. that mouse unfortunately fell extremely ill and died of that disease. It died of pneumonia. All the symptoms were the same. And then when he did a necropsy on this mouse, he cut it open, looked inside, and found this bacteria inside of it. So what's going on here? What's the difference? Well, he didn't know, genetically speaking, what was going on here. So he thought it maybe was just the capsule itself, and that kind of makes sense. Maybe it's the capsule that's killing the mouse. Maybe that's what's going on. It's the same cell that's living inside. That's not different. So it has to maybe the capsule that's killing the mouse. So what he did was very interesting, and he heated up this particular bacteria in a beaker, and he heated it up just long enough to kill them, but not boil and break them apart, but not break their cells open. So their capsules remained intact. Dead cells, but they're intact, so their capsules aren't broken. So if the capsule is what's causing the disease, if you inject that in the mouse should still develop the same symptoms, even though the bacteria is not alive. So he did that. He injected it into the mouse. The mouse never developed any symptoms. He eventually did a necropsy on the mouse and found no bacteria inside of it whatsoever. This is dead. The immune system was able just to clean up the dead bacteria and get rid of it. So nothing was found inside of that mouse whatsoever, which also makes sense. And this was the cool part here. He took this guy, which is not able to cause disease, and this guy, which is also not able to cause disease, and he mixed them together in a syringe and injected them into a mouse. That mouse then fell extremely ill with all the stereotypical symptoms and signs of pneumonia. It died. He did a necropsy on it and found this form of the bacteria living inside of that mouse. Very strange. He didn't put this bacteria there. Neither of the bacteria that he put in this mouse are able to kill the mouse. How did it die? And then how was this bacteria found inside of it? Well, he had no earthly idea what was going on here. He just figured out that there was something inside of bacteria, the something part, that was able to be transformed from bacteria to bacteria. They were able to change their shape. They were able to transform somehow. He wasn't really exactly sure how. And he just termed this something the transformation principle. So this is kind of what this concept is known as, transformation. We'll talk about what's actually going on here in just a second, a little while later, or a little while in this lecture series. But just be aware that this is called transformation, this process, where one bacteria transforms itself. This guy transforms itself into a different strain or a different form through taking up foreign DNA. And we'll talk about how this works in a little while in depth. So a couple of years later, like I said, this was in the late 20s. Now, he didn't really know what DNA was. It wasn't discovered yet. They just kind of had an idea that something was able to be transferred from bacteria to bacteria. They just didn't know what it was. So these three guys down here discovered DNA in 1944, and they also discovered that DNA was what was able to carry genetic information. So this is what was going on here. So these bacteria, they carry the genetic information for capsules inside of them. These guys right here, sorry, the smooth strain. These guys lack it. If they had it, they'd use it. They don't have it. These guys have it, so they make it. It's exactly what's going on here. These guys have one more gene that these guys lack, the gene to make a capsule. That's the only difference between the two. Frederick Griffith didn't know it was a genetic difference between them. He just thought it was something he could see. It is something you can see, but it's explained genetically. Anyway, so what happened is when he killed this bacteria, it's dead, but it's still intact. So the genes inside of this bacteria that make the capsule are still intact. They're just not inside of a living cell anymore. So what happened was he put these two bacteria, this living one that can't kill you, that lacks the capsule gene, and this dead one that can't kill you, that has a capsule gene, together in a syringe. Injected him into a mouse, and what happened was this guy started to fall apart and degrade inside of the body of that mouse, or inside of the test tube, even earlier maybe. The pieces of DNA from inside of this cell got out, the dead cells. They got out inside of the environment, floated around inside the test tube or inside of that mouse's body. What happened is the little gene for a capsule on that DNA landed on the surface of one of these smooth strain bacteria. They brought that little piece of DNA inside of their cell. I'll show you guys how that works in just a little while. They brought it inside of their cell, and then they were able to use that gene for capsules that they stole from these guys to make their own. So they transformed themselves from the rough strain to the smooth strain using a piece of DNA that they got from a dead cell, which is pretty interesting. So we'll talk about how that works in a little while. So these guys figured out what was really going on and how this experiment really worked by taking the dead killed cells and digesting them with different things that eat proteins, some that eat RNA, and some that eat DNA. And then when you add them to the rough cells, the non-capsulated cells, whichever ones develop a capsule should be the key here. So if you get rid of the proteins and the capsule still appears, it's not proteins that are causing the capsules to show up. If you get rid of the RNA and the capsules still show up, it's not RNA that's making the capsules show up. If you get rid of the DNA and then the capsules don't show up, which is exactly what they found, it's DNA that's carrying the information for capsules. And that's how these guys discovered that transformation principle was really DNA. So the term transformation stuck, but the transformation principle was replaced with DNA. So we'll talk about what transformation is in a little while. It's that concept I just went over here with the stealing DNA from a dead cell kind of thing. So inside of a bacteria, we'll talk about the nucleoid. We've talked about this one a bunch. It's also sometimes called the bacterial chromosome. It's just a little blob of DNA that sits in a little cytoplasm and just kind of floats around inside of the cell. No membrane around it. It's found in our eukaryotes. So these guys are prokaryotic. They lack that nucleus. Inside of that bacterial chromosome, that nucleoid, you're going to find all of the information that's needed for a bacteria to be a bacteria. Everything that it needs to replicate itself, everything that it needs to do to make a new generation, everything that's needed to make all the proteins, all the things that a bacteria needs to be a bacteria will be inside its genetic information here. Sometimes bacteria have things called plasmids inside of their cytoplasm as well. And plasmids are essentially just little extra small pieces of DNA that contain some genetic information. So this is what you need to be a bacteria. This tells you how to make proteins, how to replicate your cells, how to make some membranes, everything that you need to be a bacteria. This tells you how to make the enzyme that degrades penicillin, penicillinase. This tells you how to be able to survive in a little extra heat. This tells you how to be able to survive in a new type of cell. So plasmids are extra information that allow bacteria to do things. Sometimes they're useful, sometimes they're not. It just depends on the plasmid itself and the genes that are on them. But the plasmid itself is not a part of the nucleoid. It is not required for bacteria to live. They don't have to have these. Sometimes they do, sometimes they don't. And it just depends. These are not required for the bacteria species to live, or the individual bacteria can vary within the species as well. One's individual cell, while the exact same species cell right next to it does not have them. They are not part of the DNA that's required for the bacteria to survive. Now, all of this information together, the sum of the chromosomes and the plasmids, all the DNA inside of this cell is called the genome. So sometimes bacteria have mutations that occur inside of their cells. So this lecture is going to address is how bacteria change their DNA, how changes occur inside of bacteria to allow them to either become more infectious, less infectious, transmit from species to species, or things like that, how they change their DNA. So the easiest way for bacteria DNA to change, as well as pretty much every other species on the planet, is through mutation. Mutations just randomly occur in nature. Those are called spontaneous mutations. But sometimes we want to make mutations occur in a lab. And those are called induced mutations. We'll talk about those a little bit more in a second. So mutations in humans, we have hundreds of thousands of billions of cells. So if one cell has a mutation in it on the inside of my arm muscle, you'll never, ever, ever know it. You won't see it. It probably won't impact my survivability or my functionality at all. I'll never know. But a bacteria is a single-celled organism. It's prokaryotic. So if it has a mutation inside of its cell, that's going to really impact its survival. It only has one cell. So if its single cell is mutated, whatever is the problem is going to be significantly more visible in bacteria and prokaryotic organisms than in eukaryotes, things that have much more complicated or multicellular organisms. So bacteria are really good ways to study mutations. They divide very quickly. It's easy to see the mutations, and they're pretty cheap to deal with, and you don't really need a zoo whereas you would say if you studied them in monkeys or something like that, or mice, you can just study them on a petri dish. So these are the different types of mutations that can be found in nature. These are spontaneous mutations, just ones that randomly occur when the DNA has a problem. So if you want to review the lectures from earlier, the little videos that I'll post about how DNAs are read, codons and things like that. Proteins are read using 3-letter sections of DNA called codons. So if there's an error inside of the codon, the protein that is produced using that codon, it can be incorrect. So in this representation, word form, you have each single letter represents an individual DNA nucleotide. Each individual 3-letter word here represents an amino acid that's being produced, or the codon itself. So we have a normal functioning DNA gene, and it reads, the man saw the dog hit the can. 3-letter words, perfect. If this DNA is turned into RNA and then make a protein, the protein that's produced will be exactly the way that it's supposed to. It works the way it's supposed to, it functions the way it should, and this bacteria is happy with that functioning protein. Now there's one type of mutation called silent that's not on here. And a silent mutation would essentially be like changing the word can here, the letter C to a K. The man saw the dog hit the can. Can with a K. It's pronounced the same. It reads the same. For all intents and purposes, that is the same word. A silent mutation does not change the message. It doesn't change the protein that's produced. It doesn't change the mRNA message. It doesn't really matter. Now, mutations that change the proteins that are produced, or the amino acids that are in the protein chain, they matter. Those are the ones that can lead to significant problems. So the first one's something called a point mutation. The man saw the dog hit the can. Now in this case, the nucleotide G has been swapped accidentally for a different nucleotide inside of the DNA chain. Now this changes the message completely. The man saw the dot hit the can. When this occurs, the amino acid is changed. The protein that will be the mRNA message in turn is also altered, which can usually result in a different protein being formed, or a different the protein doesn't form correctly. A different amino acid's inserted, and the protein does not form correctly and probably won't function correctly. A deletion mutation occurs when an entire amino acid or a codon is accidentally removed from the DNA chain. The man saw the hit the can. Dog has been removed. An entire nucleotide has been removed, which in this case will result in an amino acid being skipped in the protein chain, which will then result in probably not so functional of a protein. You can have an insertion type of mutation. This occurs when a brand new amino acid is accidentally stuck inside of the DNA or the mRNA chain. You have a brand new 3 nucleotides that are added inside of the original DNA, which results in a brand new piece of mRNA being produced with an extra piece of amino acid information in it, and extra amino acids added to the protein, which in turn adds to the functionality of the protein, or it may cause it to not function at all. So it's an insertion mutation. And then a frame shift mutation occurs when one single nucleotide is accidentally deleted. So it's not a deletion, because a deletion occurs when the entire amino acid is removed. This occurs when one nucleotide, one letter, is accidentally deleted. Now, if you recall, DNA has to be read in letters of 3. It's called codons. It has no choice. So in this case, we got rid of the D. And when that occurred, the dog just turned into the og, it, and ek, and und. It kind of looks a little bit like German. But what happened was the D was removed. You were forced to still read in letters of 3. You just have to do with the next 3 that show up in line. The D's gone. You don't have a D anymore, so you go with the OGH, ITT, HEC, and that's just what you're stuck with. So when this occurs, obviously the protein that's going to be produced from this is going to be severely malformed and not at all what we were looking for the bacteria or the organism to function. So this is the different types of mutations. These are spontaneous mutations. These things occur all the time throughout nature, just randomly. And we'll talk about some of the ways that they occur in just a second. So sometimes you can have bacteria mutations. Sorry, I should probably go back. Spontaneous mutations just occur because DNA makes errors. It can have an error when it replicates. You could have a mistake in the protein that replicates the DNA. Anything can result in this happening. They're fairly rare. Spontaneous mutations do not occur very often. It would be bad for your DNA to have problems in it. It would mess up your ability to survive. It would mess up your ability to reproduce. Spontaneous mutations are extremely rare. 1 in 10 to the 9th cells is when they naturally occur that have anything other than silent. They're very rare. So sometimes we want to study mutations in a lab. You can induce a mutation to take place, which is the opposite of a spontaneous mutation, an induced mutation. The things that you use to induce a mutation are called mutagens, and you're probably familiar with some different types of mutagens, things that encourage mutations to take place. So chemical agents, the ones that are familiar to you are probably things like nicotine, alcohol, things that mess up the DNA inside of humans. But in bacteria, these are some very common different chemical agents that are used to alter DNA in bacteria to make mutations occur, just so we can see what happens, to study what these mutations do to bacteria, and maybe try to replicate a spontaneous mutation occurring in a lab. So you can read, if you want to, the different types of chemical agents, as well as what they do to the DNA bases and DNA chain, and how they change and alter the DNA inside of an organism. So transposons are another type of way that we can alter the DNA chain inside of a lab. So transposons are interesting little things. They're tiny little pieces of DNA that have the ability to jump out of the DNA chain that they're housed in. So just because they're stuck inside of a piece of DNA doesn't mean that they're permanently stuck there. Every other piece of DNA on the planet's pretty much stuck where it's at. These guys aren't. They're found in pretty much every single species of bacteria on the planet. And we have figured out as scientists how to use them. So what they do in bacteria is they just jump from one piece of the bacteria's DNA, the bacteria's chromosome, to a different piece. They just leave where they're at and jump to a different piece. Now, if you jump in a gene, then you turn off that gene. If you jump in a piece of non-important DNA, it doesn't matter. If you jump in a gene, then you have problems. If you split the middle of the gene in half, the protein that's going to be produced is going to be produced in 2 different halves, which means it's not going to work. So if transposon shows up in the DNA of a bacteria inside of a gene, it's going to not work. Now, we as scientists have figured out how to use this, and we can plug transposons in where we want them. We can make them jump into bacteria to turn off or on genes, which is pretty neat. And we can make them induce mutations inside of a lab. Now, these things are interesting because they can also spread from cell to cell. They can jump inside of plasmids, too. And then plasmids, as we'll talk about in a second, can be passed around through the process of conjugation, bacterial sexual reproduction. So radiation is a good way to induce mutations. You guys are probably familiar with this one. One of the most common forms of radiation damage is something called a thymine dimer. So you guys can see here we have our 2 DNA strains. And in our 2 DNA strains, we have 2 A's and 2 T's that sit right next to each other. A is bonded to T across here, and then A is bonded to T across this way as well. And that's what holds the DNA strain together. Now, if you come in contact with UV light causes the 2 different DNA strains to separate a little bit. And when they separate a little bit, the bonds break between the A's and the T's. But when the UV light leaves, the energy from the UV light's gone, the 2 DNA strains will come back together. The A bonds will form to the T's, and so on and so forth, the G's to the C's. Now, most of the time that works like it should, but sometimes what happens is when the 2 different DNA strains separate apart, and you got 2 T's right next to each other, those guys can sometimes form a bond to one another. This is called a thymine dimer. So this is changes their structure of their DNA, puts a little kink inside of the DNA. And when this is formed, it causes the DNA to improperly be able to it causes the DNA not to be properly able to be transcribed into RNA and then translated into a protein. So once these little kinks show up in the DNA, UV light, you can do this. And the same thing happens inside of your cells in a tanning bed. and when your body repair this, you produce melanin, photomelanin. So the more thymine dimers you have, the darker your skin tone comes out after a tan. So how do we select for mutagens in a lab? So you've spent all this time, all this money, all this effort trying to induce a mutation in a lab, and you finally get it happen. You finally get the mutation that you were looking for. So how do we make this guy grow? How do we separate him away from everything else? The easiest way is something called direct selection. So if you're trying to induce a mutation for, say, resistance to an antibiotic, you have a bunch of bacteria that are not resistant to an antibiotic. You're trying to make them resistant to it. So you do all of your tests, your DNA, you hit them with UV, you hit them with all kinds of stuff, radiation, trying to make them have a spontaneous mutation occur to induce that mutation. And then after you're done with all your tests, you pour them on a petri dish that contains the antibiotic that you're attempting to get them to become resistant to. Now, in this case, it's streptomycin. So you have a plate without streptomycin and a plate with streptomycin. The plate with streptomycin in it, nothing will be able to grow except for the organisms that you have induced that mutation in. So if it grows on the streptomycin plate, you know you have successfully induced your mutation. Whereas if you have the one without streptomycin, everything will grow, including the ones that are resistant to streptomycin. So that's how you get this guy away from all of the other stuff. You put a plate containing just the antibiotic that you're trying to induce a mutation for, and the ones that grow on it are resistant. So this is called direct selection. You can have indirect selection. It's pretty much the same concept, but you use two different plates. You grow up one petri dish instead of pouring these guys directly onto two different petri dishes. You pour them onto one petri dish to start with. And it's called a master plate. And then you essentially take a little piece of a toilet paper tube with some sterile velvet over it, and you make a stamp. You just press it down inside of that master plate, and then you do that same process where you put one on a plate that contains streptomycin and one that doesn't. You stamp it down, and then you incubate them. So you come back, the plate that contains ampicillin, the ones that grow on here, you'll see that won't grow, the ones that contain ampicillin will grow on the ampicillin plate, and the ones that contain ampicillin that don't contain ampicillin that aren't resistant will not grow on the ampicillin plate. So on our plate that doesn't contain ampicillin, everybody will grow once again. Since this is an identical stamp, you can look at it and go, oh, these three colonies grew on the ampicillin plate. These three guys grew on the ampicillin plate. These three guys grew on over here on the non-ampicillin plate. These two guys grew on the non-ampicillin plate, but they didn't grow over here. That's our different ones. Those are the different guys. So you have two identical plates, and the colonies that are different between them is how you tell them apart. This is indirect selection. Sometimes you can induce mutations that cause bacteria to become sensitive to conditions that they might otherwise survive. You can make them where they can't survive in high temperatures and things like that, or they can't survive in high salts, if that's a mutation that you're trying to induce. So this is a type of selection called condition lethal, where you're going to expose a bunch of normal bacteria to whatever you're wanting to try to use to induce that mutation. And then what you're going to do is you're going to expose them to a condition that should make them die. You plate them out on a plate, a replica plate, the same way you use that stamp concept. So you have two plates that are identical. And then you incubate one at 25 degrees, so the temperature that this bacteria can survive at is if it's not mutated. And then you incubate the other one at 37 degrees. So the mutated cell cannot survive at 37 degrees. The non-mutated cell will be able to survive perfectly fine. Over here, the mutated cell can survive at 25, whereas everybody else can. So the mutated cell dies over here at 37 degrees. So you look at the stamped identical plates, and the differences between the two are these colonies that you've successfully induced your mutation in. Now you can fish these little guys up and start a pure colony plate with them and do more tests with that. So how does a bacteria repair mutations? If you have a mutation occur inside of your cell, obviously that's a problem. They're not going to have a normally functioning DNA chain, normally functioning protein, so your bacteria wants to repair that stuff. So they have evolved certain enzymes and mechanisms to repair DNA damage. It can go read the DNA chain and find it and then fix that damage. So DNA polymerase will proofread the nucleotides the very first time. This is what's going to replicate your DNA in the first place. It's going to proofread from the get-go. So it's going to make sure that it doesn't make errors. Now, sometimes it does. It's not foolproof, but it makes a really good job. It does a really good job at proofreading itself, so it doesn't just make error after error after error. Your bacteria also have the ability to do something called mismatch repair, where enzymes will go along the DNA chain and find mismatched DNA bases that are not repaired correctly by DNA polymerase. So DNA polymerase accidentally pairs up a T to a T or a T to a C, something like that. It accidentally makes an error on the DNA chain. So these guys, this enzyme will go and fix that, fix the mispaired nitrogenous bases. Light repair will go in and fix that. An enzyme will go in and fix the thymine dimers, fix those that damage, fix the kink in the DNA chain. And excision repair. And this type of enzyme will essentially go in and cut out any pieces of damaged DNA or incorrectly matched DNA or anything that's not supposed to be there, and then add in the correct nucleotide sequence to fix that piece of DNA chain. Now, the problem occurs, you have all of these ways to get around mutations, to repair them and things like that. Now, the problem occurs if your body doesn't have the ability to get to all of them. Same concept of smoking a cigarette or Russian roulette kind of thing. If you smoke one cigarette, you're probably not going to have a mutation occur. If you do, your body has the ability to fix it. If you continuously smoke cigarettes, the likelihood of you having a mutation occur is going to be significantly higher. And the more you smoke, the more chances you take with it, and the higher chances of you having mutations occur in more cells. So essentially what happens is the DNA mutations occur all throughout the DNA chain. One mutation at a time, your body can repair that, or a bacteria DNA can repair that inside of its DNA. But if you have hundreds of mutations that occur over and over and over and over inside the same DNA chain, the bacteria or your own cells can't keep up with that type of damage. It can't keep up with it, and that's when you result with cancer and things like that. It can't fix itself. It can't repair itself in time, and then the mutations that occur in important places that lead to the cell not being able to correctly manage its replication. And that's cancer or the inability to replicate itself correctly at all, or it might just kill the cell and things like that. So that's how DNA mutations and things kill. Whereas they occur all the time, one or two mutations is not a huge deal. Cells can generally fix that. Multiple mutations are too much at one time. That's why the concept of radiation poisoning is a thing. You come in contact with too much radiation, you have too many DNA problems at once, and that's how you die, kind of thing. Mutations are an overwhelming game. So sometimes bacteria can transfer their DNA from one another, from one cell to one another. So we've talked about how they reproduce asexually, the concept of binary fission. They literally just make an exact copy of themselves, a clone. They replicate their nucleoid, split their cells in half, and send one copy of the nucleoid to either side. Now that occurs when bacteria are in happy environments, when everything's good, when they have all the food they need, all the nutrients that they require. Now, sometimes when the environment goes bad, these guys need to shake up their genetic information. This is called genetic recombination, shaking up the genetic information, combining two cells into one. The resulting cells that occur from that genetic recombination are termed recombinants. So we are recombinants from our parents. And bacteria that are have genetic information from two different bacteria inside of them are also called recombinants. So these are the three main ways that this occurs, and we'll talk about them in just a second. So DNA-mediated transformation, and there's that word again, transformation, the transformation principle, and this is what that is. So one cell dies, and the information that's contained inside of its DNA is released into the environment. All that DNA floats around inside of the environment. That DNA, I'll go through with the picture down here, lands on the surface of a perfectly normal bacterial cell. That perfectly normal bacterial cell will bring inside a piece of that DNA. Doesn't need both of them, they're both the same thing. It just needs one. So it'll bring in one piece, and the other piece is just degraded. So that one piece of bacterial DNA that it found on the outside can now be spliced inside of the original bacteria's DNA. So it takes its old DNA, gets rid of it, and then sticks the new piece of DNA inside. Now, every time that this cell replicates its own DNA, it's going to make a replica that contains that DNA. So one cell will have two versions. You have to make two copies of the same. One cell doesn't get it, so this guy is non-transformed. This guy is considered transformed. Every time he replicates, you have two transformed cells. This guy can't because he doesn't get a piece of it, looking at how DNA replicates, that conservative, semi-conservative method. So anyway, this could be the gene for DNA or penicillin immunity, the ability to produce penicillinase. So penicillinase floated around in the environment for a dead cell. Cell picked it up, brought it inside. Now this guy can produce penicillinase, and now he's resistant to penicillin. This guy can too. So every single cell on the planet has DNA inside of it that's useful, but not every single cell on the planet can use that DNA. A cell has to be termed competent to be able to bring inside foreign DNA. It has a little extra piece of cellular machinery added to its surface, essentially like a little fishing lure that can catch the DNA as it floats by in the environment. Not every single cell has it, and not every single species can do this. But this is a very common way for them to transfer genetic information from one cell to another. And this is how Frederick Griffith's transformation experiment worked. The gene for capsules was contained in the environment on one piece of DNA. It was brought inside the original cell's, the rough strain cell. The rough strain was then able to produce a capsule, and then any cell that it produced from then on out as it replicated was also able to produce a capsule, and then any cells that they produced were also able to produce capsules. And that's the transformation principle in a nutshell. So sometimes bacteria can be transformed by bacterioviruses, or bacteriophages, and this process is called transduction. So I mentioned on the virus lectures that viruses work a lot like postage envelopes. It doesn't really matter what's inside of the envelope as long as the postage has a stamp and an address, which is the viral capsule. It doesn't really matter what's on the inside. So if you recall how bacteriophages work, is they inject their DNA, the little bacteria's DNA is broken down. The host phage, the phage DNA that's then used to make lots of copies of the original virus. They reassemble and then burst out of the cell. Now, sometimes what can happen is the virus will inject its DNA, the little bacteriophage. The original cell's DNA is destroyed, and the bacteriophages are assembled inside of the cell. Now, this guy over here, if you notice, he didn't pick up any of his original copies of DNA. None of the copies went inside of him. He accidentally was assembled with a piece of the original cell's DNA inside. Now, once again, it doesn't matter what's inside of a virus. Only thing that matters is the capsule is intact and it's released in time, and it will go do what it's supposed to do. Now, these little guys, the green ones, once they get out of the cell, they'll go kill another bacteria. This guy, once he gets out, he will do his best to try to infect a cell. He is a virus, after all. That's what they're supposed to do. So he will go infect another cell. He will inject his DNA with it, but in this case, it's not virus DNA. It's bacterial DNA. This bacteria doesn't have its original DNA destroyed. There's no virus there. There's nothing to do that with. And then he just takes this little piece of DNA that the virus brought him, and then he uses it for himself. And then he becomes something called a transduced cell. Now, we as scientists have figured out how to use this process. I mean, we can put genetic information that we want things, bacteria, to do for us inside of a virus. And that's how we get that DNA inside of them. So this is how we use things to make insulin. So we put the gene for insulin production in a virus and then gave it to a bacteria that way. So a very cool little process using a bacteriophage virus. Streptococcus pyogenes that causes strep throat can also cause the disease scarlet fever, and it becomes infected by a bacteriophage to be able to produce a different type of toxin to cause scarlet fever. So bacteria can also have their version of sex, bacterial sex. It's called conjugation. And this is something involving the formation of a sex pelus. So this requires cell-to-cell contact. It makes sense. It is a version of sex. And this is the transformation, the ability to transfer plasmids back and forth from one another. So how you can tell that this requires cell-to-cell contact. You have a bacteria over on this form, one strain that requires a certain growth nutrient. And on this strain, you have another one that requires a different growth nutrient and a membrane in the middle. They cannot get to one another. So you take this sample out and put it on a plate, that contains the stuff that this guy can't have. They can't survive there. They obviously don't have it, and they're going to die. This guy can survive in this media, but not that. And then you put him on a different media, and they're going to die. Now you remove the middle membrane. These guys can come in contact with one another now. They can have bacterial sex conjugation. They can trade their genetic information back and forth in the form of plasmids, which allows them both to be able to survive in the presence of these different growth mediums. Now they can both eat different things. It's pretty interesting how this concept works. So how this works in simplicity, in reality, is you have a bacterial plasmid that allows these guys to form a sex pilus. This is a sex pilus over here. It's essentially just a little hollow straw that plasmids and things will be able to move between, that forms between the cell walls of bacteria. So the F plasmid is what allows this to occur, the fertility plasmid. If you have the F plasmid, you are termed an F positive, quote unquote, male cell. The F plasmid will then cause the F positive cell to develop a sex pilus. And the pilus will stretch across into the F negative cell. What will happen is that sex pilus will make a nice little hollow straw. The F plasmid will make a copy of itself. This is called the rolling circle replication. I'll post a video about that as well. And then slowly that little plasmid will replicate itself and travel across the sex pilus, and now you have two F positive cells. The sex pilus will break itself, and then the cells will go continue on doing their day business. Now, not only just the F plasmid can be carried through. Other plasmids, such as the resistance genes, R plasmids, and things like that, resistance to antibiotics, the ability to eat certain nutrients can also be carried. So pretty much any plasmid can be carried through the sex pilus. You just have to have the F plasmid to be able to make the pilus in the first place. The cool part is once you get a pilus, get that F plasmid, you can transfer all that information to a bunch of other bacteria, and you get to keep the F plasmid, which is kind of neat. And then they also get their own copy. So here's the word form of what I just went through. So genetic transfer, genetic transduction has been observed in pretty much every single living thing, but transduction using viruses is pretty rare. It's in nature, It occurs a lot in living host and species. In labs, you can use it pretty easy, but not so much in nature. So sometimes bacteria don't want to have mutations inside of them. If you don't want to have a virus infect you, that's a bad thing. The virus can kill you. It can cause bad things to happen. So viruses, as well as other types of cells, bacteria, as well as other types of cells, our cells, have developed things called restriction enzymes, which are used to target foreign DNA that comes inside of the cell. So restriction enzymes essentially look for unique sequences of DNA that are not found in their host DNA. So if I have a bunch of A's and T's and G's and C's and a very unique A, T, 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 over and over and over pattern inside of my DNA, my restriction enzymes will look for any DNA that's found within my cell that does not read that normal pattern. That normal pattern is what they expect to see. If they see something that's not that pattern, it's probably virus DNA that's attempting to hijack my cell, or maybe it's a piece of DNA that's been absorbed from a bacteria or anything like that. Who knows? It's probably bad. So restriction enzymes will look for things that are not what they're supposed to see, and they essentially will chop them into 100 tiny little pieces, get rid of that piece of DNA to prevent it from causing any problems in the first place. Now, we scientists have figured out how to use restriction enzymes, and we can use them to cut DNA in specific regions, specific ways, and things like that, to insert different genes and stuff like that. I'll post a little video on this one as well. So let's go ahead and talk about the different types of viruses and finish up this little lecture. Poliovirus. FDR, Franklin Delano Roosevelt, the president during World War II, had polio. He got it in his late 30s, early 40s, and it's very uncommon for him to be seen in his wheelchair. He wore very thick leg braces to keep himself standing upright. He wanted to put forth that show of strength, so a picture of him in his wheelchair is fairly uncommon. But he did have polio, one of the famous people that had polio. So poliomyelitis, poliovirus, this is it up here. One of the few vaccines that was super successful at eradicating a disease pretty much from the planet. Um, it's still around, but it's gone into the fact that there are only 42 people in the United States that have polio still. They contracted polio as a child, and they're still living with the after effects of polio in the sense of an iron lung kind of thing. So polio, what it does is it paralyzes the body's ability to have muscles move. So paralyzes muscle movement. So essentially what can happen is it paralyzes the ability for the diaphragm to work correctly, and that's what happens a lot. And essentially you lose the ability to breathe, and you will suffocate to death. Very unpleasant way to go. It's spread by coughing, respiratory droplets. Very, very, very interesting little virus. It is the smallest virus that has the ability to cause disease in humans. There are no yearly deaths by polio, and once again, there are only 42 people that are alive today that are in still of an iron lung. There are still cases of polio that show up from time to time. Very uncommon though. Chickenpox, Varicella zoster, also the same disease that causes, or same virus, that causes shingles. Group 1 DNA double-stranded DNA virus, spread by contact, respiratory. You come and get contact with respiratory droplets and things like that. You inhale them, put them in your fingers, get them in your nose, get them in your mouth, and things like that. This is the virus itself. You inhale them, they get inside of your lungs, and they cause these horrible little itchy splotches, to develop all over your body. Treatment by calamine lotion, just oatmeal baths and things like that to treat the itching. Acetaminophen to cool this fever, to cool the swelling, and things like that. 10 to 12 days after you come in contact with the little virus, you will start to develop symptoms, and it lasts about a week or so. This exact same virus can cause shingles when the antibody count drops. And they do have a vaccine for this, which has also been partly responsible for the shingles outbreak. You need constant re-exposure to keep your antibody counts high. Chickenpox has essentially been eradicated, so there's no re-exposure for people that have been exposed to chickenpox prior to the vaccine. So their antibody counts constantly drop. Since this particular virus can hide within the body, go dormant, if you are one of the unlucky people that has gone dormant your antibody count drops so low, it comes back with shingles. So there is a shingles vaccine to give your antibody counts a boost. And you can have your antibodies counted for this particular virus if you're concerned about shingles. I'm going to finish this one up with mononucleosis Epstein-Barr virus. This is an oncogenic virus. It can cause mutations to occur that cause cancer in the mouth. So this is also called the kissing disease. You get it very often by respiratory droplets coming in contact with saliva and things like that, inhaling them. You can get it from brushing teeth, sharing a toothbrush, like I said, sharing a toothbrush, drinking after someone. But most oftenly, it's going to be spread by kissing, things like that, coming in contact with saliva in some way, shape, or form. Very, very, very unpleasant disease. A couple of weeks of active sickness. You apparently don't want to get up and do anything, and it's just apparently horrible. Not a whole lot they can do for this. Once again, it's a virus. You can't really treat viruses. They just have to give you over-the-counter pain medication, keep your fluids replaced, and keep you just kind of feeling good, and that's about all they can do. And then once again, this thing will run its course eventually, and you'll be done with it. So this is pretty much all I got for you guys about genetic information, bacterial genetics and things like that. I'll post a video, once again, a couple of videos about a translation and transcription, DNA and things like that. So if you have any questions over this lecture, guys, please feel free to let me know. If not, have a good rest of the day.